अच्छा ये लोग बोले थे कि उसी के साथ आती है वगैरह भाई ये देखो ना दूसरी से मेरा नहीं भाई ये समझ अरे भाई अरे वो ये लोग का बात नहीं आ रहा नंबर सिम नहीं है व्हाट्सएप पर सिम डेटा कहाँ से है मेरा मेरे के नाम से ये लेकिन सिगारेट से नहीं लगे ये लोग को डाटा पड़ेगा इसको निमन से प्लान नहीं किया अबे यार तो भाई सब हो गए अच्छा से जो करती है वैसे कुछ पता नहीं है हिंदू नाम अबे वो बना इससे हम एक्सपेरिमेंट करेगा अच्छे बनेगा और फिर आगे उसमें हिम्मत नहीं होती हिंदू नाम के क्लास में क्या रहता था हिंदू नाम के क्लास में क्या रहता था निकाल देगा गेटअप समझ ना तेरे कौन हिंदू नाम सुमारा नाम अच्छी नहीं है होगा क्या आज ये कौन सा मैच का अरे मैं तो ना जब जो बोले टाइम पे भाई देखो एक ही बात देख रही है अच्छा लगे ठीक लगती है भाई समझ तो आती है तुमको भाई वो समझ ये समझ में आती है कि वो समझ में आती है ठीक से देख भाई ये भी बोल रहा है ये सही है बोल कीजिए ये एंडो को डाल दिया लेकिन मेरे समझ में तो आता है बहुत ही इसका अरे मेरा आदत हो गया प्रोसेस की करने को दिक्कत नहीं है मैं देखो क्या रहा हूं सर्टिफिकेट जहां अपन नहीं होता तो ये मेरी ब्रांड है वापस अरे होमवर्क की क्या है तो ऐसे क्या होता है कांड क्या तो दे दिया क्लास की जगह है हिंदू मनीष है अरे हिंदू सुरेश है अबे होमवर्क निकाल ले उसका नहीं अगर पेट को से ये तभी क्या है बहुत अच्छा इसका राशन ये लास्ट सम से जो के नहीं लास्ट सम में तो लास्ट होगी नहीं अच्छा पापा ने भी बैठे हैं
पार्टी का ब्रेक है ना पार्टी का ब्रेक है ना मैं आता हूँ
एक्चुअली आपको बात करने हाँ लेकिन हमें कोई ना बात करने को रहता है बात करने क्या होगा इसका अगर आप मोबाइल है तेरे पास ज़रा कर भाई एक मिनट अरे भाई वो थोड़ी सी अच्छी है अलग नाम है मैं उसने देखा था डायग्राम अरे अभी तो अपने स्ट्रक्चर पे काम करते हैं हाँ वो भी वही हमने स्टार्ट किया और फिर एक्शन दे अरे चाहते हाँ वो पढ़ते थोड़ी है सीधे आगे कितना लेके स्टेट में चलो अरे एक्स्ट्रा इस पेज कर सकता है इस नहीं के अरे तो मैम तो हम कुछ उसको इतना 10-15 पॉइंट भी मार्क करवा रहे हैं लेकिन इसको थोड़ा सा मत जा हमको बोला था नहीं लास्ट स्टार्ट में पूरा कोई थी ना मुझे नहीं लास्ट में हमारा बहुत बड़ा वो काटा था भाई हम लोग तो वैक्सीन पढ़ते हुए पता ब्रेक की पढ़ते नहीं हमें 1.5 मार्क चले गए नहीं वो मार्क किया मैंने नहीं करा था कौन हिंदू में क्या करती थी क्या उसको आंसर 1 मार्क भाई वेल वो ना टैब वो है मार्क करना है रॉन्ग हो गया वो नहीं जाना तो भाई फुल नहीं मारता लेकिन अक्षर बोल दो डेढ़ मार्क से क्या फर्क पड़ता भाई डॉट चाहिए सुरक्षा आगे पीछे से ना दो जाओ अरे गिर गया गाड़ी का एक्सीडेंट हुआ था क्या कैसे हुआ गाड़ी में भाई मैंने ठोक दिया अरे मैं कल उसका मैंने देखा पर इतना बार चला था हां और इधर से कल मैंने उसको देखा स्कूल में हां एक्सीडेंट आया था हां तब भी हुआ लेकिन ना सुनना स्कूल में मैंने देखा उसको इतना लग गया था उसके पहले दिन वैक्सीन लेने के पहले दिन उसको देखा था चल रहा था आराम से अपने बहन के साथ ऐसे ऐसे आराम से अरे ठीक है ये कभी तुम कर सकते अरे को मारा क्या बताएगा तो अपना नहीं बताते हुए मेरा नाम है अरे संजय ग्राउंड में ना उधर ही मिलना अरे साथ में क्या देर करता प्रैक्टिस करता है ग्राउंड में हां प्रैक्टिस करता है मिलान में प्रैक्टिस बोल के आवाज कोई गेम नहीं अरे डायरेक्ट मैच ही खेला था मैं देखने को नहीं मैच खेलता रनिंग को कुछ नहीं बोल तो 500 को खर्च किया मुझे तो रनिंग को करना है मेरा स्कूल में क्रिकेट करो 500 रुपए लेते हैं मेरी स्पीच पे ना ना बाउंस है ना कुछ भी नहीं है सीधा मार्क्स को क्या लेकर खेल रहे हो पट्टी बोला कोई डेढ़ पॉइंट ना हो ग्रास भी ठीक बात है वो ग्रास भी ठीक बात है हां पिच नहीं है ये लगता है स्कूल में हमने पिच मार कर अबे आजकल लोग किसके लिए लेते हैं भाई कुछ नहीं मुझे खेलने में क्या चाहिए पैसा जो बुलाने 1000 रुपए मत जा मैं तो 10 रुपए तो खेलते हां सीनियर से देखते हुए नहीं भाई हमारे में एक भी सीट है आपका लोग खत्म आ रहा है इसको भी हम बताने आए अच्छा लगता है सब लोग वैसे ही क्रिकेट भी ना अरे वो बाजू वाली क्लास में कौन है? ए में ना ए। अरे इधर सी। हाँ सी सब के उधर भी कॉमर्स है ना। सही बात है। भाई तो भी कितना इंटीग्रेट सैचुरेशन
अरे हम तो थोड़ा ब्राइट है। अब लेकिन तुम्हारे रहते हैं हमारा मंडे का तो चला का वैक्सीनेशन की चक्कर में हमारा ये चला गया वीक में।
अरे बीओ की क्लास में ना। नहीं मेरे क्लास में
बीओ डी में है ना। डी में तो हमारे साथ होगा।
हम लोग मंडे को ना वर्सेस रहता है हमारा बीओ।
अरे बीओ अरे नहीं।
अरे कितने
वर्सेस नहीं होता हमारी ऊपर अपने बाहर ना तो उस दिन सब्सटीट्यूशन था।
नहीं अरे अपना टिकट अच्छा उनका सब्सटीट्यूशन उनका सब्सटीट्यूशन
ये वाला अपने नहीं खेलने वाले क्योंकि वो मूव है
वो तो अरे हम दोनों को डाला था बस ये देखा गया है
अरे हम लोग क्या कर रहे हैं
तो ये उन्होंने बहुत अच्छे देश ने मारा था
तो
तुम्हें तो था
टीम में क्या करेंगे
रिलेशन में अकेले थे पूरी टीम में बीओ में तो क्या ही कर रहा था क्या बोल रहा है
योगा क्लियर
अबे तो मैं उसके भी करते चलते
मतलब वो मिला को भी
मिला
Good evening. So, present we were studying structure of cell. Did we finish drawing the diagram?
Yes, ma'am.
Animal cell. How long did we finish? Show me. Animal cell get up.
And now the difference between prokaryotic and
That the introductory part is over. We have already drawn the diagram of the animal cell. Today, what are we going to learn? We are going to learn the cell organelles one after the other. Straight away, we go into the cell organelles. Now, to talk about the cell organelles, what are we studying? You are new? Today, first class?
No, third class.
No. My lecture
Yes.
first. Whatever has been already over, just go through in their
I wrote the notes, but I will draw the diagram.
that you can do it. So, cell organelles meaning already, if you remember at the start of this chapter, when I was introducing the word cell, already I told you what are the components of cell. If you remember, already I told you what are the components of cell. Again, if I write, what are the components of cell? We generally write components of cell would be what? Cytoplasm hai. What else? Cell membrane hai. Also known as plasma membrane or nucleus. These are the major components of the cell. Now that I talk about the cytoplasm, and we are going to study the topic cell organelles today. Cell organelles are all present in the cytoplasm. Then what are the cell organelles that we are going to study today? There are a lot of cell parts, just like the parts of our body. If I say examples like the brain for a distinct function, heart for a distinct function, the lungs for a distinct function, the bones for a distinct function, the liver for a distinct function, like this, no? So if I'm talking about that, our body has got so many different parts, and every single part is for a distinct function. That way, if I'm talking about the cell also has got different such parts, and every single part for a distinct function. So that is what we are talking about as cell organelles. We say organs of the body, and the parts of the cell, organelles. Then what are the organelles? We will be talking about mitochondria. We will be the other day I already showed you the diagram we have drawn also. Mitochondria. Look at the mitochondria in your diagram now. We have the endoplasmic reticulum. What else? We have the Golgi apparatus, right? We have the Golgi apparatus. What else? We have the ribosomes. Then we have the lysosomes. Can you name next? Centrioles. What else in the plants, animals, plant diagram we haven't drawn? Did I miss any? Mitochondria, nucleus I did not write. Though we are writing here nucleus as a component, nucleus is also a cell organelle. Okay? So these are the cell organelles for the animal cell that we have to learn. Now, of course, I have to add two more, the vacuole and the plastids. But we haven't drawn the diagram of the plant cell, so I'm not writing it because these two are organelles of the plant cells, not in the animal cells. So we'll go by the sequence more or less. Let us start with the first cell organelle, mitochondria. Now, as we start looking about the cell organelles, try to understand what exactly. Actually, this cell organelle will be try studying. It's a big concept. But according to your syllabus, we'll try to stick to your syllabus and learn only certain parameters that you are supposed to understand. So first of all, we will, okay, let me just start from here. The cell organelles, and then what are we to study? Let us make a column or parameter to study the structure and the function. Very clearly, you should remember this way. Organelle, structure, function. I start with mitochondria. Why? Because it is the most important. Most important. Then we'll look at the cell membrane also. That's also there. So let us start with the mitochondria. How does it look like, the mitochondria? The structure, if you have to draw the mitochondria, it is a double wall structure like this. Yes or no? A double wall structure like this. And on this double wall structure, what do we get to see is there are some studs as if, some knob-like structures as if all over. There are some studs or knob-like structures all over this way, and they have a specific name and a function. We are talking about the mitochondria, so separate apart from all these structures, a very, very distinctive feature about the mitochondria that you have to remember is the mitochondria has got its own DNA. Now, this DNA that the mitochondria has, is it very long? Yes, it is very long and it always remains coiled and single. Okay, what else about the structure? This much at least you have to remember. What is this? The outer membrane. I want you all to draw this diagram. The outer membrane.
We have to make this whole table.
No, you don't have to make the table as such. You will understand what I explain you, and when I give you questions, you will write. I will finish every organelle or one or two organelle, and then I'll give you a set of 3-4 questions. So, you have to important points only you will remember, because the same thing is there in your textbook. So there's no need of writing down what I write exactly on the board. It is simply a repetition. जो मैं अभी लिखने वाली हूँ almost सब तुम्हारे textbook में है. Outer membrane. Now this is the inner membrane. This is the inner membrane. The inner membrane has folds like this. Can you see folds or finger-like structures? Folds. So what are the folds of the inner membrane known as? The folds of the inner membrane is known as crista or cristae. Say suppose if I write two of them together. Cristae, singular crista. What are these dot-like structures? The dot-like structures are known as F0 particle. The dot-like structure, F0 particle. And what is this DNA known as? Mitochondrial DNA. We write this way, mtDNA, mitochondrial DNA, small m, small p. And what about the matrix? Let me make the diagram a bit more beautiful, but you cannot. You have to draw only with the pencil. No coloring, no sketch, no shades with any crayon, sketch pen, nothing. Only pencil. When you are asked to draw the diagram, number one, diagram should be a little bit away from the margin, almost in the center. Like the other day I told you, not exactly in the center, a bit away from the margin. Half a page. In this chapter, 3 to 4 diagrams are important. Diagram should be at least. Now this diagram, if you draw the diagram of the cell, that should be almost 8 to 9 lines. When you are drawing it as a line, single line notebook. Now that you draw the structure of the mitochondria, it should be at least about 4 to 5 lines. That big the diagram should be. हाँ, मुझे पता था कि तुमने जरूर छोटा ड्रॉ किया होगा। हाँ, मम्मी ने बोला ना, यही नोटबुक और 5 साल चलाना है। ऐसे बोला मम्मी ने। तो these type of small diagrams, at least this type of small diagrams जो individual organelles draw करो, एकदम 2 लाइन में draw नहीं करना है, not that too small, but 4 to 5 lines में करना है. But if it is the cell, then 8 to 9 lines, 8 to 10 lines में करना है, because so many things inside you have to draw. Make it fast. At least 3 to 4 organelles we should finish today. mtDNA. mtDNA का मतलब है mitochondrial DNA. This is called mitochondrial DNA. शॉर्ट में mtDNA. Cristae, C-R-I-S-T-A-E. T-A-E. Singular is Crista, I told you. T-A-B, right? But since I have labeled 2 together, so it will be Cristae. T-A-E. इसमें F0 particle. F0. Subscript 0. F subscript 0. ऐसे नहीं लिखोगे. You will not write this way. You will write this way. गिरा, बैक गिरा, फिर उसके बाद तुम गिरोगे. इतना हैवी क्यों है? 2 तो बुक लेके आना है, एक मैथ्स का और एक बायो का. Done. Make it fast. Last one more minute I can give you. Jaldi karo. Mitochondrial DNA short form में small m small t DNA. This chapter is over in school.
No ma'am, almost later.
Almost.
Later was.
यहाँ तक ही पहुँचे. वो आगे से होते हैं.
ये ना मतलब थोड़ा 2-3 ऑर्गेनल्स हुआ. ये पहले 5 कर दिया आपने. हाँ ठीक है.
कितने कर लिए? 4.
एक बात सेल वॉल बात करते हैं तो
आज मैं 5 ऑर्गेनल करके ही जाऊँ.
और ये बैठ
तो आगे मत बोल. हाँ.
आगे बीच में पहले कार्डियो फिर बाद में इधर कोई से ठीक करवा देते फिर एक और से कार्डियो हो जाता है फिर वो एक और वापस आता है फिर वो भी एक और कार्डियो हो जाता है
दिन पढ़ाने का मूड को पढ़ाएगी पढ़ाने का मूड नहीं को पढ़ाएगी हम स्कूल में ऐसे ही करते हैं यहाँ पे वो ऑप्शन नहीं है पढ़ाना ही पड़ेगा. चलो we'll finish with this. First is mitochondria, a very important organelle. Structure याद रखना है. This is the structure. So structure में what are the important parts to remember? First of all, it has 2 membranes. So that's the reason we will say this is double walled. It is double walled. Two double walled because it has got one outer membrane and inner membrane. No? What next? The inner membrane is folded. So what are the folds of the inner membrane known as? Cristae. What are the folds of the inner membrane known as? The folds of the inner membrane are known as cristae. So this comes as a definition. What are the folds of the inner membrane of mitochondria known as? The folds of the inner membrane of the mitochondria is known as cristae, and the inner membrane is always folded. So अभी inner membrane जो folded है, can you tell me what might be the significance? Significance क्या है? Significance is to increase the surface area of the membrane. अगर folded नहीं होता, अगर it would have been the same oval membrane like the outer membrane, उसका area कम होता. And अब जब ये folded है, तो इसका area ज़्यादा हो जाता. And on top of that, it has got some small knob-like structures, and these knob-like structures are known as what? The knob-like structures are known as F0 particle. We already wrote the F0 particles. Now, these F0 particles are the sites for ATP synthesis. F0 particles are the sites for ATP synthesis. What is the full form of ATP? Adenosine triphosphate. ये क्या है? Adenosine triphosphate. It is the energy molecule produced by the mitochondria. Mitochondria produces energy molecule. Where does it form? The energy molecules are getting formed in the F0 particle, जो एक knob-like structure है. So if I draw this bit bigger, समझो, if I am drawing one of these knobs bit bigger, तो वो कैसे दिखेगा? It will be something like this that I am drawing, जहाँ पे इस तरह के कुछ छोटे-छोटे जैसे ना mushroom जैसे कुछ structures है, छोटे-छोटे mushroom जैसे structures है. And this is what we are talking about as the knobs of the cristae. And the knobs of the cristae are actually detailed structures where the process of ATP synthesis takes place. काफी सारे chemical reactions takes place. Chemical reactions takes place which help in the formation of ATP. What is ATP? Adenosine triphosphate. You will be asked questions. Name the energy currency of the cell. What is the energy currency of the cell? ATP. Where is it formed? Mitochondria. State the location of ATP formation in the mitochondria. Cristae. F0 particle अगर याद भी नहीं रहा, cristae याद रखना है. The cristae of the mitochondria. So ये हो गया structural part. Apart from that, now we have to understand one more very important concept about the structure of the mitochondria. Mitochondria is known as a semi-autonomous organelle. Mitochondria is a semi-autonomous organelle. What is the meaning of the word autonomous? English में autonomous का मतलब पता है क्या? What is the meaning of autonomous? हाँ? अरे नहीं ना, वो खाली वो gadgets और machines से याद आता है हाँ? Autonomous. What is the meaning of autonomous? Anything which is almost independent. North Point School is a semi-autonomous. मतलब ये fully अपने आप decide नहीं कर सकता, इसके पीछे और भी trustee है, etc. government है. So when I talk about any organization, school है, college है, university है, or any individual, say suppose you all autonomous. When I talk about yourself, you are also like the mitochondria, semi-autonomous. You are still not autonomous. I can be, okay, I am more or less autonomous. I can take my own decisions what to do. But you, maybe not yet. You are still not mature enough to decide. You have to depend on your mother or father. You have to depend not only because of money, but you are not that mature to decide correctly for you, right? That's why. So when we are talking about what is the meaning of autonomous? Fully independent. Semi-autonomous, partly independent. Okay. Now, why are we talking about these words, independent and not independent? Other organelles apart from mitochondria, if I speak, endoplasmic reticulum, Golgi, ribosomes, etc., they are fully dependent on the nucleus to take any function. What function should they perform? How much enzyme, protein, etc. should they release? Fully dependent on nucleus. But the mitochondria has got its own DNA which can release so many enzymes or which can give commands to release so many enzymes. Mitochondria has got its own DNA. Is there any other organelle which has its own DNA? No other organelle apart from the mitochondria has got its own DNA. हाँ, plastids में भी होता है, but plastids तो plant cells में होता है. Right now I'm not talking about plant cells. So in the animal cells, if I talk about mitochondria, is one sole organelle जिसके पास अपना DNA भी होता है, and that's the reason it can take its own functions. It can decide for its own functions, which enzymes, which proteins to form. हाँ, थोड़ा बहुत तो nucleus पे dependent है ही. That's why semi-autonomous. हाँ बोलो.
What exactly is DNA?
DNA is just a chemical molecule, chemical compound.
But what is it significant?
DNA has got all the different, I should say, information. This is something like some codes here. And these codes are going to give commands for the formation of proteins. हमारे body में there are a need of almost about 400 to 500 different type of proteins, so that each and every function of our body happens. हमारे eye के functions, brain के functions, stomach के functions, pancreas के functions, bone के functions, all are dependent on variety of chemical reactions. So for those reactions, how much of the protein, what enzyme, when, ये सारे commands DNA देता है.
So in this DNA specific.
अभी इसमें तो mitochondria में खुद का भी DNA है. Generally, the nucleus is known as the director of the cell. Huh? So when I talk about the meaning director, you understand any structure or a body or a person who gives instructions, commands. So हमारे cell के अंदर भी there is an organelle known as nucleus which gives commands to every organelle. You have to do this, you have to do this, you have to do this, you have to do this. Like that. So mitochondria, is it dependent on the nucleus? Yes, but partly, because it has got its own DNA also. Something like after, maybe now you all are in the 9th standard, maybe after another 10 years, 12 years, you start earning money, you get good jobs and you are earning enough for yourself. So at that time, what happens? You have, say suppose you have rented a house for your own, you want to buy some new good clothes, you want to buy your own vehicle. So you have to, right? Oh, you are smiling at him, or he is smiling there.
I just.
Now, when all these things happen in your life, what happens? Are you not going to discuss with your parents? कि मुझे BMW खरीदना है. So your father says, okay, go for it. So you have your own independence, plus you take the advice of your father. He has got his own independence, but still he is partly dependent on the nucleus. Semi-autonomous. Why? You get questions. Why is mitochondria known as a semi-autonomous organelle? Because it has its own DNA. It has its own DNA.
Ma'am,
Bolo.
nucleus is autonomous, then?
Nucleus is?
Autonomous.
न्यूक्लियस इज फुल्ली ऑटोनोमस क्योंकि न्यूक्लियस का फंक्शन कोई और ऑर्गेनल डायरेक्ट नहीं करता है. न्यूक्लियस itself has got a lot of enzymes and proteins to direct other organelles outside the nucleus in the cytoplasm. Has its own DNA to synthesize variety of enzymes. So why is DNA known to be semi-autonomous? Because of the presence of its own DNA, and it is partly dependent on nucleus. It is partly dependent on nucleus. Now, let us write down some important functions of mitochondria. The first is, I will write the first function of mitochondria is cellular oxidation. Cellular oxidation. What is the meaning of cellular oxidation? Cellular oxidation and release of energy. Something like, if I talk about a cell in our body, every cell in our body has got adequate amount of glucose. Now, we eat carbohydrate food. No, we eat different types of food. Even from fruits, there are carbohydrates entering our body from rice and from bread and from roti and from all these carbohydrates entering our body. If I talk about the variety of fat substances that we eat, that also can get converted into carbohydrate to produce energy. The proteins, maximally we eat a lot of protein in our food. That also partly can get converted into carbohydrate after the process of digestion. So when we talk about every cell in our body, there is adequate amount of glucose. Now this glucose has to get broken down to release energy. So what is basically necessary by the way of respiration? Oxygen should enter the cells. No, there is the role of oxygen. There is the role of lungs there. There is the role of RBC there. There is the role of hemoglobin there. No, we'll learn later in the chapter respiration. So nutrition से glucose जाएगा in the cell. Respiration से oxygen आएगा cell में. And then what? As a result of this reaction, what will be formed? Carbon dioxide तो है ही, but we are interested about energy. तो हमारे body cells में adequate amount of glucose, oxygen का आना बहुत जरूरी है, and that is a reaction which takes place. अभी तो मैंने simple एक concept दिखाया है. So that is a major reaction which takes place in our cells of the body, and as a result, energy gets released. Now see, now that I am talking, बहुत energy की जरूरत है. Now that I am standing, energy की जरूरत है. But are you spending energy when you are sitting? अरे हाँ. If you don't have energy, then you will lie down. एकदम relax, एकदम गिर जाओगे. Are you drooping? No, you are not drooping. You can sit properly with your muscles and backbone erect. क्यों? Because you have got energy adequate in your body. You are able to write, you are able to draw, you are able to ask, you are able to talk. You have got adequate amount of energy. अगर energy नहीं होती, you would have felt tired, you would have felt exhausted. हाँ? अब सो जाते हो तुम मेरे क्लास में. So now when we are talking about cellular oxidation, so ये जो process, ये माइटोकॉन्ड्रिया के अंदर ही, और ये जो प्रोसेस है, ये वो वाले सेल्स में पॉसिबल है, वो वाले ऑर्गेनिजम्स में ही पॉसिबल है which have got mitochondria. Meaning, if I talk about a bacteria, prokaryotic, eukaryotic पढ़ा है ना? Bacteria is a prokaryotic or a eukaryotic cell, prokaryotic. तो बैक्टीरिया के अंदर तो enough amount of mitochondria नहीं होगा. So is the bacterial cell able to undergo this process? No, not at all. Not at all. और भी बहुत सारे ऐसे सिंपल ऑर्गेनिजम्स हैं which come under the category of prokaryotic. या तो माइटोकॉन्ड्रिया नंबर इज वेरी लेस. सिंपल ऑर्गेनिज्म माइटोकॉन्ड्रिया का नंबर अगर बहुत लो हुआ तो एनर्जी प्रोडक्शन भी बहुत लो. So this process of oxidation and release of energy is very much associated with the presence of mitochondria in the cell.
Some that is called mitochondria is called as
powerhouse. तो ये जो एनर्जी है ये किस फॉर्म में रिलीज होता है? इट रिलीजेस एनर्जी इन द फॉर्म ऑफ ATP releases energy in the form of ATP. So ATP synthesis सबसे important function ये another very important function is ATP synthesis. ATP synthesis is another important function of mitochondria. What is the full form of ATP we just now learned? Triphosphate. We ask energy currency of the cell, what it is? It's answer is ATP. What is the third function of mitochondria? Can synthesize large number of enzymes. So this mitochondria can also can synthesize a large number of enzymes. Respiratory enzymes we have both सकते हैं. Okay, overall enzymes, they can produce a large number of enzymes, they can release energy in the form of ATP, and they can also help in the process of oxidation, a process which very much helps in the release of energy. What is it? Oxygen and glucose to react, and then carbon dioxide and energy to be formed. So you will write, when you are asked functions of mitochondria, mitochondria helps in the process of cellular oxidation. Mitochondria releases energy in the form of ATP. Mitochondria synthesizes a variety of enzymes and proteins. Proteins we accept when I wrote enzymes. The word proteins as it is coming through. या तो proteins word add भी कर सकते हो. It releases a variety of enzymes and proteins. So this is about mitochondria to understand. Next, let us talk about the plasma membrane. Let's talk about the next concept, plasma membrane. Now, after the cell organelle, I go to the concept of the membranes, the limiting membranes of the cell. The plasma membrane, when I talk about, let us try understanding. Now, when I talk about the plasma membrane, what am I trying to understand? Say, suppose this is my cell and I have the nucleus here. So, plasma membrane means I am trying to magnify this part of the cell and try to understand. So, when I magnify this part to understand, I get to see that the structure is almost like this. The structure is almost like this with two layers of fats, almost this way, two layers of fats. What are these layers of fats known as? These layers of fats are known as lipids. Lipids. What is this layer known as? Lipids. Two layers of fats, lipid molecules. In between lipid molecules, what is there? In between the lipid molecules are some proteins this way. In between lipid molecules are some proteins this way. Are some proteins this way. Are some proteins fully embedded, or some proteins this way. Okay. So when I talk about the structure of the membrane, the plasma membrane, I get to see that this plasma membrane has got two types of molecules very prominent. What is it? Some protein molecules and lipid molecules. So whenever we are talking about that, the basic structural aspect of the membrane, the basic structural aspect of the cell membrane, what it is? The basic structural aspect is composition. First of all, we try to understand what is the composition. Composition is, it is made up of lipoprotein, meaning it is made up of two layers of lipids and a layer of proteins. But the proteins may be arranged in three different ways. How? One, which is partially embedded, where you can see a part of these protein molecules from the upper surface. Some protein molecules are fully embedded through the width. Fully embedded मतलब अगर ये मेरा दो layer है, some proteins are partly embedded ताकि मुझे ऊपर से half दिख रहा है. Some proteins are fully spanning the width. ऊपर से भी थोड़ा दिख रहा है. अगर मैं नीचे का structure magnify करूँ तो नीचे से भी दिख रहा है. Some proteins are embedded from the top in such a way कि वो नीचे पूरा दब गया है, ऊपर से नहीं दिख रहा. So it may be any of these arrangements, but yes, proteins are a major arrangement. Now, this type of arrangement is basically known as the fluid mosaic structure. The fluid mosaic structure. ऐसा क्यों? Because when I talk about the lipid molecules, are they solid molecules? No, they are not solid. They are, the lipid molecules are almost like gel, semi-fluid. So they have got a semi-fluid consistency. They have got a semi-fluid consistency. It's not solid. And in between the proteins, no, just suspended. The proteins are suspended and they form the firmness of the entire membrane that way. Fluid mosaic structure. Why the word mosaic? Because when we talk about the mosaic floor देखा है ना, where there is the white background, the white cement background is there, and then there will be the colored marble chips के designs बने रहते हैं. Colored marble chips के designs बने रहते हैं. देखा है ना, ऐसे वाले floors बहुत common है. In especially the old buildings, you will come across the flooring is that way. In mostly the hotels and घरों में भी होता है, but in our houses it will not be those elaborate design, but it will be just the marble chips. No, we get to see on the white cemented floor a marble chips जो कभी orange color के छोटे-छोटे marble chips दिखते हैं ना, orange color, yellow color, brown color, red color के chips दिखते हैं. These type of floors are called mosaic floors. And elaborate designs भी होता है, like in those palaces and the hotels we go at. So now ये जो mosaic floor है, यहाँ पे भी जो construction is more or less the same. White layer of cement रहता है, and then the marble chips are spread out. And जब polish किया जाता है, some marble chips पूरा अंदर चला जाता है, some of them are fully embedded, some of them are partially embedded, and some ऊपर वाले layers पे रह जाता है. So the proteins are also similarly arranged this way. Hence, it is known as fluid mosaic. So what remains as the composition? Lipoprotein. Lipoprotein. What is the composition of the plasma membrane? Lipoprotein. Next is what? The plasma membrane is, when we talk about the features structurally, the plasma membrane is semi-permeable. The plasma membrane is semi-permeable. What is the meaning of semi-permeable? Semi-permeable means I may talk about a cell, any particular cell of our body where we understand that the cell is producing certain molecules, certain proteins. The cell is producing almost about, this cell is producing almost about 2-3 types of proteins, 2-3 types of proteins. But the cell is allowing the movement of only this outside. And this type of molecules, no, the cell does not allow the movement outside. At the same time, this type of cell is able to take in certain molecules. It can take in certain molecules, but it will not take in these molecules. Is it able to take this? No, but it can take this. So what is the meaning of this? That every cell has a preference for taking in molecules or giving out molecules. For example, I will talk about the alveolus that we have in our body, which is also a cell. And these membranes, if I talk about, yes, these are also semi-permeable, where we understand this can majorly allow the movement of oxygen in one way and the movement of carbon dioxide in the other way. And there are many other molecules to talk about this way, where we understand whichever membrane you talk about, bone cells, liver cells, pancreatic cells, muscle cells, cells of the stomach, etc., etc. Every cell has got a preference for certain molecules to go out and for certain molecules to come in. Any molecule cannot go out and any molecule cannot come in. That is what is known as the semi-permeable nature. Permeable matlab movement is possible. Semi matlab partly, not fully. Semi-permeable, selectively permeable. Matlab certain few molecules can come in and certain few molecules can go out. Not that all molecules can go in and all molecules can come in. So these are the two properties of cell membrane that you have to remember. What is the other name for cell membrane? The other name for cell membrane is plasma membrane. The word plasma is also from the word fluid. It is not solid. It is semi-fluid. Plasma states when we say, no? Plasma, semi-fluid structure it is. What are the functions, let us see. What are the functions of the plasma membrane or the cell membrane? At least 2 to 3 functions you have to remember. What are the functions? The first function that we talk about of the plasma membrane or the cell membrane. The first function of the cell membrane holds the cell contents. Holds the cell contents. Yes or no? Does it hold the cell contents? Does it allow all the cell contents to remain inside? Yes. अगर plasma membrane या cell membrane नहीं रहता, then the cytoplasm and the organelles would have leaked out, no? Would have come out, would have got spread. And if I'm talking about the liver, say suppose if I'm talking about the liver, which is now made up of so many cells and the organelles inside, just think for any organ or a tissue, if the cell does not have membrane, क्या होगा? All the contents will come out and they will all mix, no? They'll all mix. Are the cells now able to perform any definite function at this time? No. All getting mixed and it's just a mess inside the tissues and organs. Holds the cell contents. What else? Gives a shape and framework. Gives a shape and framework to the cell and the tissue formed. So from the structure of the cell, the shape of the tissue also depends. The structure of the cell, what is the structure of the cell? Now say suppose when I talk about the nerve cells, what is the shape I showed you in the first lecture? Shape of the cell. What is the shape of the nerve cell?
They are long.
Long. What is the shape of the muscle cell? Cylindrical. What is the shape of the RBCs?
They are long.
Almost spherical. What is the shape of the stomach cells? Almost rectangular. So whenever we talk about the shape of the cell, that also more or less decides what should be the shape of the tissue and the organ that it gives rise to. No? Gives the shape and framework to the cell and the tissue that is forming from it. And the third and the most important is the process of transportation. Transportation of molecules, ions, and enzymes in and out of the cell. Transportation of molecules and ions and enzymes in and out of the cell. So that is what is the major function of the cell when we are talking about the major function of the cell. I'm sorry, the cell membrane. When I talk about transportation, just now we were talking about the cell, the selective permeable nature also. So from the selective permeable nature also, you are supposed to understand transportation. Say suppose, if I talk about the stomach cells, the stomach cells are releasing enzymes. No? So if I show you this way, that this is the stomach, and let me say that this is the stomach cell, and this stomach cell is releasing certain enzymes. What is it? What is this? Inside the stomach cell, certain enzymes form. They are coming out of the cell. Transportation, coming out. At the same time, there are certain cells. If I talk about any particular organelle where I would understand that from one cell the molecules are moving to the other cell. Is this possible? That is also possible. And from another group of cell molecules are moving into, that is also there, no? So from one cell to another, substances and molecules are constantly moving from one cell to another. Molecules are moving from one tissue to another, molecules are moving. If we don't understand this, then how would the molecules from the intestine move into the blood? How? Cell to cell movement, no? How will the molecules of glucose and amino acid move into our bones for the process of growth? Migration, no? Transportation, no? Cell to cell transportation is happening. So in our body, this type of cell to cell transportation from one part of the body, one tissue to the other is very common. So this function. Take one question now. Write one question now. Number one. Explain the semi-permeable nature of plasma membrane with an example. Explain the semi-permeable nature of plasma membrane with an example. Explain semi-permeable nature of plasma membrane with an example. Answer.
Semi-permeable nature.
Explain the semi-permeable nature of plasma membrane. Explain the semi-permeable nature of plasma membrane. Answer लिखो.
With an example.
With an example. I will give you with an example. Because the plasma membrane or cell membrane, the plasma membrane or cell membrane is semi-permeable. The plasma membrane, take that question. These are important concepts that you have to remember. The plasma membrane or semi-permeable, the plasma membrane or cell membrane is semi-permeable or selectively permeable. That is semi-permeable or selectively permeable. That is certain molecules can enter or leave the cell. Certain molecules can enter or leave the cell. That is, certain molecules can enter or leave the cell. For example, certain molecules can enter or leave, L-E-A-V-E, leave the cell. Certain molecules can enter or leave the cell. Can you tell me any example? आ रहा है कुछ याद में? Cells where movement is happening in and out. अच्छा, हाँ, वो तो semi-permeable membrane का example है. Alveolus. We have discussed about alveolus. अच्छा, it is right. An example about the semi-permeable nature of the cell membrane. Write down. Certain molecules such as oxygen, mineral nutrients or digested nutrients such as glucose, passes through the cells of the villi. Villi are structures in the small intestine, finger-like structures. Passes through the cells of the villi to enter the bloodstream. Okay, so if I talk about cells of the villi, this is a very good example to remember. You will study also in detail. Cells of the villi, which are nothing but cells of the intestine, After the process of digestion, they cross the semi-permeable membrane to enter into the blood vessels. But is it that all molecules can cross? No, certain proteins cannot. But glucose can, sodium, potassium can, iron, mineral, nutrients, some can, but all cannot. Okay? So semi-permeable nature of the membrane. Next is, we'll just start with this concept. We'll not be able to finish today, but we'll do it the next class. Next organelle that we talk about is endoplasmic reticulum. Endoplasmic reticulum. For this diagram not needed at all. Only two types you have to understand. Structurally, the two types. Let us just finish with this. What are the structural types of the endoplasmic reticulum? The first type is known as rough endoplasmic reticulum. Rough endoplasmic reticulum. In short, RER. And second is smooth endoplasmic reticulum. Smooth endoplasmic reticulum. In short, SER. The difference being is structurally, let us complete. When I talk about the rough endoplasmic reticulum, it is the association of the endoplasmic reticulum with another cell organelle known as ribosomes. Remember, check your diagram that you have drawn. No? On the surface of the endoplasmic reticulum, how did you draw? There is another organelle present associated like, no, this like small granules over. That is the rough endoplasmic reticulum. But if I have to draw the smooth endoplasmic reticulum, I will just draw this way. No granular structure. So what it is? It is only the endoplasmic reticulum and no ribosomes attached. It is only the endoplasmic reticulum and no ribosomes attached. So that is when we are talking about the difference between endoplasmic reticulum rough and smooth. For rough, ribosomes are attached. For smooth, no ribosomes. Functions, three basic functions. We'll discuss the next class and continue with the other organelles. For this, you don't need any diagram. Okay. Yeah.
आपको कुल सा मिला तरीके से है सब मिला है एंड साइंस और एसएसटी मिला होगा ना ये भी है ना नहीं है हो गया ये नहीं कह सकता
तो क्या पूरा एक दिन छुट्टी नहीं है मतलब लेकिन एक ही क्लास में चल
Good evening. Yes, what was the homework? 1.3.
homework exercise student homework exercise student
Two and three go on. Write the lowest rationalizing factor. Here root 24, 3 root 7. Given equation, you multiply this, write this in the form of a squared minus b squared. I'm so sorry. What absent? Really? Absent. I didn't miss you. See the denominator there, it will become root 5 the whole square minus root 2 the whole square. नीचे हम लोग रैशनलाइज इसके लिए करते हैं ताकि वो a squared minus b squared के फॉर्म में बन जाए. So what is root 5 squared? 5. Root 2 squared तो 5 - 2 आपका नीचे 3 हो जाएगा, ऊपर आपको मिलेगा 3 into √5 minus √2, फिर आपका ऊपर का 3 नीचे का 3 कैंसिल हो जाएगा। तो ये छोड़ दिया क्या? Even you have a doubt on the last one? This time you also have a doubt on the last one also. So the rationalizing factor is this part you understood, rationalizing factor. Now, after this, if you see, this has become a plus b into a plus b. What is a plus b into a plus b? A plus b the whole square. नहीं पहचानने में आते कोई टेंशन नहीं, नहीं ध्यान में आता फॉर्मूला कोई टेंशन नहीं। हम क्या करेंगे, अगर फॉर्मूला नहीं यूज करते हम तो हम ब्रैकेट खोलेंगे, √3 into √3 plus 1 ये तो आता है ना, ये तो हम बचपन से करते आ रहे। जरूरी नहीं कि मुझे फॉर्मूला ही लगानी है। अभी √3 into √3 3 plus √3 plus √3 plus 1 √3 into √3 plus √3 minus √3 minus 1, ये और ये आपका कैंसिल हो जाएगा। 3 plus 1 4, apple plus apple 2 apples, 3 minus 1 2, अब ये 2 और 2 कैंसिल हो जाएगा, सच्ची होगा नहीं 2 और 4 होगा ना? अरे रुद्रा इसका आंसर बोल। नहीं 3 और 3 कैंसिल नहीं होगा, अभी तो बोला तुमने, समझा? जब बीच में प्लस या माइनस साइन आता है वो कैंसिल नहीं होता है। This is your final answer. Do you understand? हाँ जल्दी किया मैंने, मुझे अपनी हैंडराइटिंग अच्छी लगती है इसलिए किया।
जब किया था वो भी अगर आप गलत कैसे कैंसिल करके आपको
क्यों कैंसिल किया?
अगर आप गलत था
तूने क्या किया मैं बता रहा हूँ, तूने भी सही किया ना बिल्कुल। तूने क्या किया है, तूने ऐसा लिखा है वो भी बेटर है ना? तूने फॉर्मूला यूज किया है। अभी a² plus 2ab plus b², तो बात तो एक ही है ना। जैसे करो फॉर्मूला मेथड से करो अच्छा है, नहीं तो आता है फॉर्मूला नॉर्मल करो। सही था, सही था। ये √6 कैसे है दिखाना है, कैसे है ये √6
सर? √6
कैसे है, कैसे है
सर? 24 को अगर 6 से मल्टीप्लाई करें तो 1.4 मिलता है उसका।
लेकिन तेरे को उससे ध्यान में कैसे
सर मैंने मल्टीप्लाई करके 1 से सेकंड
तूने अपना टाइम वेस्ट किया, तूने अपना टाइम वेस्ट
Yes, sir.
वो प्रिमिटिव मेथड है, ये लॉजिकल मेथड है, समझ गए? एग्जाम में वो देख पाए बच्चे को समझाएंगे नहीं समझा। आई एम नॉट इंटरेस्टेड इन द आंसर, इज दैट क्लियर? हाँ लिखा ये गलत बच्चे a plus b का होल स्क्वायर सब गलत है नींद में रहता है अपनी दुनिया में रहता है तो कोई पड़ेगा नींद में रहता है मेरा सब दूसरा है दूसरा कोई दूसरा ड्रीम करता है ऐसे रहता है ये सब क्लास में मतलब तो अटेंशन
सर मुझे ये वाला नहीं सीखना सर
कौन सा
ये वाला
ये वाला मतलब क्या ये वाला
क्लास वर्क का
कौन सा
वो क्लास वर्क का क्वेश्चन 3 वाला
मैंने किया क्या समझा
नहीं
तो फिर कैसे समझाए
ऐसा आपने बोला ना जो आपने एक्सप्लेन शुरू की थी और वो वाला कंप्लीट कर अब फिर जो करते करते नहीं कर
मैंने बोला कंप्लीट
आज
मैं ऐसा क्यों करेगा
नहीं तो मेरी टाइम खत्म हो गया
थोड़ी गलत बोला
मैं कभी बोला
बोला ना
हाँ बोला था मैंने कंप्लीट किया
ठीक है नहीं कौन सा नहीं समझा कौन सा नहीं समझा तेरे को
ये वाला
करेंगे हाँ मतलब कि और क्वेश्चन कीजिए मैं हाँ आईपीएल में गया हाँ तो आशीर्वाद कुछ नहीं समझेगा
सर क्रिकेटर बनना है उसका
मैं नहीं बनना
तो फिर ऐसे तो तेरा काम पानी में नहाने का ड्रिंक्स के रंग आता तू कौन से स्कूल पे अब तुम्हारा टीम कहाँ तक पहुंचा है
सर
तो कितने बजे शाम को भी खेलते हैं तुमको तो 5:30 का तो सर बोला नहीं मेरा क्लास है क्लास भूल गया
वेरी गुड
तो अभी एक सम क्या बताओ एंड हमने क्लास में कहां तक किया था उसका
सर क्लास में थर्ड
थर्ड ठीक है तो अभी आप लोगों को मैं 8th बताता हूं ठीक है और 4th 5th 6th 7th जो लोगों ने नहीं किया वो लोग खुद घर पे ट्राई करेंगे हाँ
सर होगा क्या हो
उन्हें किया उसने किया लेकिन बाकी लोगों को करने देना तो हम 8th करते हैं पहले हाँ क्वेश्चन नंबर 3 सब क्वेश्चन नंबर 8 rationalize the denominator 6√3 minus 3√5 by 5√3 minus 3√5. Obviously, the rationalizing factor of a minus b is a plus b. So I am going to multiply both numerator and denominator अभी देखो इसमें I have no choice but to open the bracket, both the brackets in the numerator. So I am opening 6√3 getting multiplied by this entire bracket minus getting multiplied by this entire ये यहाँ तो ओपन कर सकते हो यहाँ तो a minus b into a plus b कर सकते हो क्या करना चाहते हो
सर ये वाला
ऐसे वाला कोई बात नहीं जो करना है whatever you are comfortable with, we will do. I made a mistake in copying this up. नीचे right है ठीक है ओके यहां तक समझे सब लोग अभी ये देखो 6 5 30 √3 into √3 3 plus sign it 6 3s are 18 √3 into √5 √15 minus into plus minus 3 5s are 15 √5 into √3 √15 minus into plus minus 3 3s are 9 √5 into √5 5 5 5s are 25 √3 into √3 3 5 3s are 15 √3 into √5 √15 minus into plus minus 3 5s are 15 √5 into √3 √15 minus into plus minus 3 3s are 9 √5 into √5 5. Have you understood till here?
Yes.
Now see, these two fellows can get cancelled. 33s are 90. 9 5s are 45. 18 mangoes minus 15 mangoes. 3 mangoes. Did you understand this? Upon 75 minus 45. Any doubts? Now 90 minus 45. 45. 75 minus 45. 30. Can I take 3 common in the numerator? Ones are, tens are. My answer is 15 plus √15. You don't understand. Clear? You can do it by any method. You can have it here. Do it by any method, but go slow. Don't be in a hurry. सचिन, वो सचिन समझा? बैटिंग करता है ना? Over everybody. This was still, I think, question number we had done.
Yes.
Sum number was 3.
Yes.
Now we move on to sum number 4. I am going to do in sum number 4, sub question number 3. 1 and 2 you all will try yourself now. First I am doing sum number 3, so please pay attention. The heading says find the values of A and B in each of the following. We will find out the values of A and B by equating the like terms on both the sides. I repeat, by equating the like terms on both the sides. So watch my step in sub question number 3. It says root 3 plus 4 root 2 the whole upon 3 root 2 plus 5 root 3 is equal to A minus B root 3. मेरे हिसाब से मैं काम करता हूं कि जो पहले है ना, मैं ये सेकंड वाला कर लेता हूं, थोड़ा इजी वाला पहले कर लेता हूं, so that you will not get confused in the other one. ये थोड़ा बड़ा है, पहले मैं ये दिखाता हूं आपको सेकंड वाला. Only first one you can try yourself. Now I have to rationalize this denominator. The rationalizing factor of root 3 minus root 2 is root 3 plus root 2. Is this clear? Now I'll open the bracket. अभी सीखो डिनोमिनेटर में a minus b into a plus b है, so that will become a minus b, a plus b is a squared minus b squared. यहां तक समझे? 3 root 3 plus 3 root 2 upon 3 minus 2. What is 3 minus 2? 1. 1 है denominator में, so I need not write it again. I will say therefore 3 root 3 plus 3 root 2 is equal to a root 3 minus b root 2. Now equating the two sides, equating the two sides का मतलब है ये √3 है ना, इधर भी √3 है, इधर √3 के आगे क्या है, इधर √3 के आगे क्या है, हमें a और b की वैल्यूज चाहिए, सिर्फ तो जवाब देगा। a √2 है, ये भी √2 है, ये क्या है, हाँ क्या है, सिर्फ b है, अरे माइनस b है, माइनस b is 3, therefore b is -3, उनको साइन के साथ देखना है। b अगर आप 3 लिखोगे तो आपको रॉन्ग मिलेगा। जब आप इक्वेट करते हैं तो वी इक्वेट डोंकी विद डोंकी एंड मंकी विद मंकी। इज दिस क्लियर? तो b का साइन इधर माइनस है, इधर प्लस है, √3 है, तो आप उसको b को -3 लिखो। If you write +3 here in this case, you will get 0. Is this clear? Yes. Now I will show you this sum. तो अभी इसका rationalizing factor is 3√2 minus 5√3 है कि नहीं? अब ध्यान देना बच्चों कि ये इसकी तरह नहीं है a + b a - b, ये a - b और a + b था, सेम था, लेकिन इधर सॉरी 3√2 मैंने वही सेम लिख दिया, 5√3 अपॉन 3√2 माइनस 5√3 है ना। तो अभी ये a + b a - b है ना, लेकिन ऊपर a + b a - b नहीं है, तो ऊपर हम लोगों को ब्रैकेट्स ओपन करना पड़ेगा। ओपन करते हैं, √3 into 3√2 minus 5√3 plus 4√2 into 3√2 minus 5√3 whole upon 3√2 squared minus 5√3 squared is equal to, ये पूरा कंटिन्यूअस अपने को साथ में लेके चलना है। अभी हम ब्रैकेट खोलते हैं, 3√6 minus 5 √3 into √3 plus 4 3s are 12, √2 into √2 2, plus into minus minus 5 4s are 20, √2 into √3 √6 whole upon 3 का स्क्वायर √2 का स्क्वायर 5 का स्क्वायर √3 का स्क्वायर। ध्यान देना बच्चों, -12 2s are -15, 3 minus 20 minus 17 √6 4 बार समझाएगा। 24 ले लिया, -15 ले लिया, 3 apples, -20 apples, -17 apples.
Sorry, apples क्या?
सवाल एक पूछा, अरे मैं जस्ट एग्जांपल ले रहा हूँ, √6 बोलने की जगह पे मतलब सेम क्वांटिटी है ना, सेम। तब से मैं समझ रहा हूँ, उसको एप्पल समझ रहा है, मैंगो समझ रहा है, मैं जब एप्पल मैंगो बोलता हूँ कि मेरी तरफ देखता है।
पता ही नहीं तो मुझे।
मतलब मैं एक एग्जांपल ले रहा हूँ, सेम क्वांटिटी है ना, similar quantities can be added. Supposing I will add the children, I will say there are 5 boys and 2 girls in the class. Can I say there are 3 girls here? You might. And I'll say there are 3 girls here, and there is one Sachin there. Can I say that? No. वो इसका मैं एग्जांपल लेता हूँ, एप्पल मैंगो को किसे नहीं लेगा, ठीक है, √6 Okay. So 3√6 minus 20√6 is -17√6. अच्छा वो पूछ लिया।
तो मैं मुझसे पूछ रहा था, ऐसा पूरा एग्जांपल है।
Okay. कोई नहीं। 9 2s are 18. 25 3s are 75. अभी बच्चों ध्यान देना, बहुत ध्यान देना है। 24 minus 15. अभी थोड़ी अकल बोला, 24 minus 15. 9. Minus 17√6 upon minus 57. गंदे नंबर आए ना, इसलिए मैंने पहले वो वाला किया। पहले ये वाला करता तो ये मेरे से एप्पल पूछने के लायक भी नहीं बचता, समझा? ये घर जाके मैंगो ही खाता। क्लियर? यहां तक समझा? अब ध्यान देना, बहुत ध्यान देना। इधर ऐसा नेल पॉलिश लगना, इधर 2 टर्म है और ये है ना एक ही टर्म है, ठीक है, बीच में माइनस एक ही टर्म बन गया क्योंकि नीचे डिनोमिनेटर है। तो अभी ये इधर 2 टर्म है और हमें ए और बी फाइंड करना है, तो इसको भी हम 2 टर्म बनाते हैं। तो देखो वहां हम रखेंगे -9 by 57 minus becomes plus. अच्छा ठीक है, ये समझ लेना। अच्छा ये 9 है, ये नीचे -57 है, झूठ तो नहीं बोला मैं, पीछे माइनस साइन है, 17√6 है अपॉन -57 है, इज इक्वल टू a minus b√3. Can I equate the 2 sides? Can I equate the 2 sides? Why no? No. Because there is √3 here, there is √6. There is apple here, there is mango here. So I cannot equate them. I can equate apple with apple, mango with mango. शांति रख। मैं अभी एप्पल मैंगो पे पहुंच गया हूं। 3 3s are 9. 19 3s are 57. ध्यान देना. In maths, we generally write negative sign in the numerator. So minus 3 by 19 17 root 2 by minus 57 into root 3 is equal to a minus b root 3. विमान हमने इसको इसकी तरह बनाया क्योंकि मुझे इक्वेट करके a और b का वैल्यूज फाइंड करना है. Did you understand? यहां तक समझे सब लोग? Therefore, a is minus 3 by 19. चलेगा ना, क्या कर सकते? इधर भी डिनोमिनेटर है ना? है, लिख नहीं रहा अपने को, लेकिन है. And b is minus 17 root 2 by 57. Have you all understood this?
Yes,
तो मैंने आप लोग को दो डिफरेंट टाइप ऑफ सम्स कराए. दिस वाज द फर्स्ट वन, सेकंड है, ये सेकंड है और ये थर्ड है. तो फर्स्ट वाला आप लोग खुद करो. इनमें तो मास्टर बन गए 2 साल. Yes, finished. Huh?
So इसमें तो इसमें भी बहुत अच्छा था so इसमें तो स्टूडेंट में करता है तो उससे अच्छे से बहुत कॉम्प्लिकेटेड है ये
भाई लेकिन तुमको फॉर्मूला जिसको नहीं आता उसका क्या करेंगे फिर आप?
इसलिए फॉर्मूला के साथ लीजिए ऐसे क्या आते हैं
हाँ मैं बस समझ लो आइडेंटिफाई नहीं कर पाऊँ उसको क्या है plus 2 and what is a minus in the whole square wrong is it
and minus
अच्छा
समझा तो wrong जाने से अच्छा better to be safe है कि नहीं सचो
चलो स्कूल में वाटर वो
हाँ
है
सर उसको टॉपिक से
वो बोलते खेलने नहीं जाते तो बोलते हैं क्रिकेट खेलने जाते हैं मतलब क्या होएगा ब्रेक्स लगा रहेगा अपने वो अपना 2.5 मिनट्स का ब्रेक होता है ना खेलने में उनका ऐसा करते हैं
What is this problem?
रुद्रा जी हो गया आप गुम है अभी तक दिखाइए अरे बॉस आप रॉन्ग कॉपी करोगे ये नीचे माइनस था ना उसका रैशनलाइजिंग फैक्टर प्लस होगा ना तो ये बोलो योरसेल्फ इतना नॉन बचा आई कैन डू n नंबर ऑफ सम नंबर को तो जब तक आप नहीं करोगे
So got the answer.
बताइए दो क्या मेरा a
A7
b
4
मेरा तुझे मिला क्या ऐसा क्या मिला क्या
सर इसको फॉरवर्ड
आपने रूट 2 स्क्वायर क्यों लिखा ये तो 2 है ना तो 2 स्क्वायर होना चाहिए ना तभी तो जाएगा 4 होता है अब ये मुझे बताओ वापस करो वापस आपने नीचे भी रूट 2 दे दिया है इसके लिए एक्चुअली आपको रूट 2 माइनस 3 मिलता है 2 माइनस 3 शुड बी माइनस 1 प्लीज डू इट अगेन नाउ right continuously every step a plus b root right
मैं पर लिखा नहीं
वापस करो सीखना है अभी अपने को चॉइस नहीं
मैं तुम्हें भी करना भूल गया था पहले
इसलिए तो बोला ना फॉर्मूला नहीं आता है तो फंस जाओगे।
चलो ब्रैकेट तो बहुत कॉम्प्लिकेटेड
नहीं आ रहा ना। इसका rationalizing factor in the numerator we've got a plus b into a plus b, which is a plus b the whole square. In the denominator we've got a squared minus b squared. ठीक है बच्चे? Now a squared plus b squared plus 2ab a squared minus b squared root 3 का square 3. 7 plus 4 root 3. ये तो 1 बन जाते हैं, लिखते नहीं हम लोग वापस। देखो a is 7, b is 4 ऐसा। होमवर्क के लिए classwork question number 5.
सर वैसे ही करना है।
सोच के करो। Classwork question number 5 and homework question number 4 and 5. Homework is classwork question number 5 and homework question number 4 and 5. Is this clear? So we'll stop there. Okay. Thank you very much. Bye.
Thank you, sir. आज तो 800 हो गया है। जब भी 2 पीरियड होता है ना आजकल वो एक्स्ट्रा There's no more inside that we remove this outside and
Really, no?
अंग्रेजी में ही भाई वो सब तो वो सर्च करना पड़ता है ना क्वेश्चन डाल के तो वो भी नहीं पूरा टेक्स्ट बुक जैसे सारे क्वेश्चन टाइप में है हाँ नहीं हाँ वही है शायद वो बायजूस का ना उसको मत इसका रॉन्ग है उसको
Vani, am I audible?
Hello, sir.
Am I audible to you?
Yes, sir.
Okay. In last lecture, we drawn the diagram and everything.
Yes, sir.
Just give me a minute. मला उतरायला लागेल. खाली ना खाली कुठला आयडिया वरती बसून करतो ती फिरते काम कर त्याला पाठी घे Funny, am I audible?
Sorry, sir.
No audible to you?
So I can't clearly hear you.
Give me the okay. Am I audible now?
Yes, sir.
Okay, fine. I think we have completed. Last lecture was cell membrane.
Yes, sir.
Yadani, what have you completed last?
So we drew the diagram for the plant animal cell.
In terms of your osmosis, diffusion सब पढ़ाया था.
So osmosis, no, sir. You had taught me till active transport and passive transport.
Okay, fine. Dhwani, I would rather go with the next, that is, वो पूरा cell part जो है is in terms of osmosis. When you come physically, that time we will see. कल वो सामने सामने करोगे तो समझ में आ जाता है. So we will skip that part for time being, and we do a easier part, that is, आपके पास volume है?
Sorry.
UT volume.
So I can't hear you clearly.
I'm not audible to you.
So in between you cut off.
एक मिनट यार. Unstable network. Audible to you or not?
Yes, sir. Now I can hear you.
Voice cut होके आ रहा है?
Yes, sir. Sometimes in between.
Okay, just wait there. Unstable voice, unstable network. Other than the unstable network.
पूर्ण नहीं होता.
When am I audible properly?
Yes, sir.
Bunny, what we will do is, we will time being के लिए अपन cell membrane और nucleus skip कर लेते हैं.
Okay.
You have a volume with you?
Yes.
ये है.
Yes, sir. I do have it.
Okay, fine. So we will start with the page number 85. पहले का पूरा part skip कर दिया. When we come in the physical class, I will explain that part. Don't worry.
Okay.
So we go with the B part. Page number 85 पे लिखा हुआ है cell organelles. We said what are cell organelles? All those structures which are present within the cell, which have definite structure and function, is what I mean by cell organelles.
Okay.
That first one they given you is endoplasmic reticulum. हमने diagram draw किया था endoplasmic reticulum का.
Yes, sir.
One end of the ER is joining the nucleus, and other is joining either the cell membrane or it lies within the cell. एक तो cell के अंदर है या from nucleus to the cell membrane तक है. Now there are two types of ERs formed. One is the another is the smooth endoplasmic reticulum.
So I could in between. So the first one.
What do you need?
So you had told that there were two types of organelles.
Yeah, two types of ER. Endoplasmic reticulum दो type के.
Yes, sir.
One is RER and one is SER.
Yes, sir. Smooth endoplasmic reticulum and rough endoplasmic.
Rough endoplasmic. Now, what is the difference between rough endoplasmic and smooth endoplasmic?
And the rough endoplasmic, we have the ribosomes stuck to it.
Good. A very simple way to understand this is अपन आइसक्रीम खाते हैं, एक प्लेन आइसक्रीम, वैनिला या जो भी प्लेन, nothing added into it, so there is absolutely plain. But in that same ice cream now you put raisins या फिर tutti frutti आता है ना, so
Yes.
क्या rough बन जाएगा appearance.
Yes.
So when the ribosomes are attached to here, उसका appearance rough हो जाता है, so we say it as rough endoplasmic reticulum. And they are devoid of ribosomes, then I will say it as smooth endoplasmic reticulum. Is it clear?
Yes. Sir.
Now, for us to understand वहाँ पे differentiate लिखा हुआ है. RER ribosomes attached, SER ribosomes are not attached to the surface.
Yes. Sir.
Ribosomes are present now. What is the function of ribosomes? Across any cell, चाहे वो plant cell हो, animal cell हो, चाहे any cell हो, there's only one function. That is protein synthesis. So what is the function of RER? Protein synthesis.
So can you repeat again? What is the function of ribosomes?
Function of ribosomes is protein synthesis.
So I can't hear you properly.
Protein synthesis.
Protein synthesis. Okay.
Whereas that of SCR is lipid synthesis.
So 1 second. So can you repeat what is of SCR?
Lipid synthesis.
So lipid synthesis.
मतलब fat synthesis.
So your voices are going around. I can't hear you properly.
Wait, come back. But just give me a minute.
Yes, sir. Sir, if you use the headphones, I could probably hear you better. Yes, sir. So the meeting ID in the sense the
Zoom link.
okay, sir. So I'll just send you the number then once.
Fine for me now. Just give me 5 minutes. I need to log in.
Yes, sir.
Okay, by the time you can go through that part, RER, SER, differential functions.
Yes, sir.
Okay, we continue. Am I audible? Taking too much time.
Sir.
is the last number 79653.
Am I audible now?
Yes, sir.
Okay, fine. I think now it is properly.
Yes.
Okay. Actually, very specifically, RER, rough endoplasmic reticulum. Okay. Now we go on to the part that is smooth endoplasm. Smooth is what? One without ribosomes. So when I say one without ribosomes, function is lipid synthesis. So we have RER, protein synthesis. We have SER, lipid synthesis. इतना समझ में आया? Okay. Now, where is RER seen? Normally, RER seen in the plasma cells. What are plasma cells? Blood के अंदर type of cells होते हैं जिनमें ये दिखाई देते हैं. Whereas SER is seen in more of the specifically liver cells or fat cells. Now, majorly it is found in the liver cells. So SER more is found in the liver cells or fat cells. Am I clear?
Sir, so will this come in exam, like the question they will ask this question that where is SCR present or where is RER present?
RER is present in the plasma cells. Okay. SER is present in the liver cells. Will do. If you write fat cells, also will do. Not an issue. दोनों right है.
Okay, sir.
Okay. Now try to understand the next part. अब next page पे turn करोगे तो functions लिखे हुए. Functions of what? Functions of ER. Okay. Adhani, try to understand this. Function of ER अलग है. Function of RER अलग है. Function of SER अलग है. When I say function of SER and RER together is what I mean by function of ER. ER दोनों मिला के होता है. SER अलग हुआ, RER अलग हुआ. Am I clear?
Yes, sir.
Okay. Now, first function, membrane biogenesis. What is RER producing?
Sir, RER is
RER क्या बना रहा है?
lipid synthesis.
RER is protein synthesis. Ribosomes जहाँ पे भी होंगे protein ही होगा.
Okay.
Okay. What is your SER producing?
Sir, lipid synthesis.
Okay. अब दोनों मिला के क्या बनेगा?
It's a
Lipoproteins.
lipoprotein.
What is lipoprotein? We said it is the cell membrane.
Yes,
So
sir.
cell membrane lipoproteins है, and lipids is produced by SER, protein is produced by RER. So together we say it as membrane biogenesis. Membrane मतलब cell membrane, bio मतलब biological, genesis मतलब formation. So biological formation of cell membrane is done by SER and RER together. That is ER. Is it clear?
Yes, sir. Sir, so SCR and RER on membrane biogenesis.
Yeah, membrane biogenesis is function of together ER. दोनों का मिला के ER.
Sir, I didn't understand this.
See, if I take, like suppose if I take example, you are brother and sister.
Yes, sir.
तो ब्रदर का एक काम है प्रोटीन बनाना, एससीआर का एक काम है लिपिड बनाना, पर दोनों मिला के क्या काम कर रहे हैं एज अ फैमिली, दोनों मिला के यू आर फॉर्मिंग द लिपिड दैट इज मेम्ब्रेन पार्ट. सो व्हेन आई आस्क यू अ क्वेश्चन व्हाट इज द फंक्शन ऑफ ईआर, तो ईआर में यू विल राइट ऑल दिस फंक्शन व्हिच आर गिवन इन द वॉल्यूम. membrane biogenesis, transportation, framework, deoxification, ये सब कुछ लिखोगे. But if I ask you specific question, what is the function of RER? तब can you write membrane biogenesis?
Yes, sir.
No. Membrane biogenesis दोनों का है.
अच्छा.
अकेले का नहीं है ना.
ER and RER.
It is only RER. Is RER producing cell membrane?
Yes, sir. 1 second. So it is RER is producing plasma cell.
RER is producing only protein, madam.
Okay.
सिर्फ प्रोटीन बना रहा है. SER सिर्फ लिपिड बना रहा है. अगर इन जनरल पूछा गया क्या ER क्या कर रहा है, तो वो दोनों बना रहा है ना. ER में RER भी और SER भी है, तो दोनों साथ में क्या बना रहा है? मेम्ब्रेन बायोजेनेसिस.
Okay.
Is it clear?
Yes, sir.
आया ना?
Yes, sir.
Okay. Now, second function. Some proteins and lipid function as enzymes and hormones. So RER के जो function proteins होते हैं, वो enzymes या hormone ऐसे काम करते हैं. So that is also one part which is there. Then third function, transportation. Now whatever material needs to be transported via nucleus, nucleus से जो transport होता है within the cytoplasm of a cell or outside the cell, it is going via ER only. इसलिए अपने diagram में भी draw किया था. Starts from the nucleus, ends in the cell membrane. Yeah, starts with the nucleus and end in the cell part itself. So that is where transportation का channels create करता है, and through these channels it is passing into the cell or outside the cell. Am I clear?
Yes, sir.
Third, framework. Now what is framework? Okay, can you see the classroom behind us?
Yes, sir.
यहाँ पे एक पिलर है, दिखाई दे रहा है?
Yes, sir.
अगर मैं पिलर को निकाल दूं तो क्या होगा?
So it will be hollow or so it will be open.
I remove the pillars of the room, तो क्या होगा?
So the building will fall.
Good. The ceiling will collapse. The building will fall. Sir, वो support कर रहा है ना?
Yes.
Okay. Now imagine in this diagram जो अपन ने draw किया है, center में nucleus है और आजू बाजू में ER है. कहीं ना कहीं ER touch कर रहा है cell membrane को. So if I remove the ER part of it, what will happen? The cell membrane will collapse onto the nucleus. वो withstand कर रहा है because of the ER. So that is why it provides framework to the cell. Okay. All the activities, all the organelles which are there, they are restricted in their position because of the part that is your ER. Am I clear?
Yes, sir.
Just give me a second. Can you see the whiteboard? Whiteboard दिखाई दे रहा है ध्वनि?
Yes.
Okay. Now you can see over here. ये आपका cell membrane है.
Yes, sir.
This is the nucleus part. And इस पूरे part में ये है endoplasmic reticulum. मैं rough draw कर रहा हूँ.
Yes, sir.
मान लो ये 4 endoplasmic reticulum. So first is what they have said, transportation. So whatever nucleus need to transport within or outside the cell. So यहाँ से material इधर जाता है cell के अंदर, या from here it goes like this outside the cell. So that is the function of transportation. Second function is framework. It is a cytoplasmic framework providing a surface for all the biochemical activities. So if I remove this part, what will happen? ये पूरा नीचे collapse हो जाएगा. Okay. this membrane would come collapsing onto the nucleus. Yeah, this membrane would come collapsing onto the nucleus. दूसरी चीज, there is an organelle over here. Any organelle, you take mitochondria, Golgi body, कुछ भी ले लो. So can the organelle move on this side over here or over here? No, because बीच में ER है. Okay? So it restricts the organ to its place. It provides a framework to the cell. That is this part where the enzyme activities and all the things takes place. So that is why, yeah, bolo Dhoni.
Yes, sir. So none of the things move, which like the mitochondria, the vacuoles don't move from one place to another.
It would move, but within that restricted area. अगर जैसे इस जगह पे है, तो ये यहाँ से यहाँ पे move करेंगे. But can the thing move from here to here?
Yes, sir. No, sir. Because
यहाँ पे इधर ER है ना.
Yes, sir.
So here, this organelle can move only in this zone. इतने ही part में move कर सकता है.
Okay.
Yeah. So it allows a certain movement, but not complete as such. Then another important function of ER, detoxification. SER present in liver cell vertebrates help in detoxifying many poisons and rings. Now, to make it more clearer, just a simple way to understand it. If I draw a line draw कर देता हूँ, and I say this is RER and this is SER. So what is the function of RER? We said.
Sir, the function of RER is protein synthesis.
Synthesis. What is the function of SER?
Lipid synthesis.
Lipid synthesis. Okay. Apart from this, what is the function of RER? RER का common function, there are some common functions also. जैसे कि ये framework के लिए काम करता है.
Yes, sir.
Same goes for here also. This also is acting as a framework. फिर दोनों के लिए वापस एक common function है.
Transportation.
Transportation. मैंने दोनों के बीच में लिख दिया, दोनों के लिए common है.
Yes, sir.
But apart from this, there is one special function, okay, which is दोनों का combined function है. Membrane biogenesis. But there is one exclusive function of smooth endoplasmic reticulum.
Detox.
Detoxify करता है because it is present mostly in the liver cells, and we all know liver is a detoxifying unit of your body. So if in the exam they ask you what is the function of RER, तो क्या लिखोगे?
The function of RER is that it has protein synthesis. It does
protein synthesis, दूसरा framework, तीसरा transportation. ये तीन function लिखोगे.
Yes.
Am I clear?
Yes, sir.
Yes. अगर smooth endoplasmic reticulum को पूछा तो क्या लिखोगे? Lipid synthesis, framework और transportation और additional detoxification. चार function.
Yes.
Is it clear?
Yes, sir.
But if they ask you give the function of ER, RER का नहीं, SER का नहीं, दोनों का combined पूछा गया. So what you will say it as? You will write framework, transportation, membrane biogenesis, बस तीन ही. Detoxification, yes, combined function में दोनों का आता है because SER का function तो उसके लिए भी लागू हो जाता है. So detoxification also.
Yes, sir.
All these are the functions of ER. Am I clear?
Yes, sir.
Okay. ER में you can see the structures which are there. ER are long elongated tube-like structures. Now, if I take this tube-like structures and try to compress it, उसको ऐसा press कर दो, तो अपन ने वो बचपन में देखा ना balloon से खेलते थे. So I try to press the balloon in between, side से balloon will bulge out. The bulge out takes place from the side. So from the ER is formed the next organelle known as Golgi apparatus. So ER ही Golgi apparatus को बनाता है. Golgi apparatus was first discovered by Camillo Golgi. इसके लिए उसको नाम दिया गया Golgi apparatus या Golgi bodies. Am I clear?
Sir, I had one.
बोलो.
In the animal cell, how many cell SCR and RER can be there?
How many ना?
Yes, sir.
You cannot count. It depends upon cell to cell. ऐसा कोई specific नहीं है.
Okay.
जैसे कि SER मतलब liver cells will find there are more SER in the liver cells. अगर वही चीज आप मसल सेल्स में देखोगे तो मसल्स आर नथिंग बट प्रोटीन्स.
Yes, sir.
मसल्स प्रोटीन है तो वहां पे आपको RER ज्यादा दिखेगा.
Yes, sir.
Okay. Now Golgi apparatus, see the second point. A stack of short flattened membrane bounded sacs called as vesicles. अगर वो ossical मतलब circular shape में है Golgi bodies, then we say it as vesicles. You can see in the diagram. Golgi bodies के side में अपन ने ball बनाया. That round balls are known as vesicles. Okay. इसमें से बहुत बड़ा हो जाता है तो उसको बोलते हैं vacuoles. These stacks are called as cisterns. What are cisterns? The long elongated tubes जो से compress हो गए उनको बोलते हैं cisterns. Diagram में ध्यान देना. Cisterns वहाँ पे लिखा हुआ है. Okay. Then membranes of Golgi apparatus are connection with the membranes of ER and thus form a complex cellular membrane system. And इसको अपन higher level पे बोलते हैं endomembrane system. What is endomembrane system? Endo मतलब within, membrane मतलब the membrane part, system grouped together. ER से बनता है Golgi body, Golgi body से बनता है lysosomes और vesicles और vacuole-like structures. तो कहीं ना कहीं all of them एक structure से सब कुछ बनते हैं, इसके लिए उसको endomembrane system कहा जाता है. Is it clear to you?
Yes, sir. I had a doubt.
Yeah.
What was the other name for Golgi bodies?
Golgi bodies in plants is known as dictyosomes.
So what?
Writing on the
Sir,
Yeah.
what is this exactly?
you can see on the screen dictyosomes लिखा है मैंने.
Yes, sir. But what is it exactly?
They are nothing but Golgi bodies only. पर plant में उनको अलग नाम से जाना जाता है. In plants they are known as dictyosomes. In animals they are known as Golgi bodies. Is it clear?
Yes, sir.
They are one and the same term, my dear.
Yes,
दोनों
sir.
एक ही है. Only plants में उनको अपन dictyosomes कहते हैं. Animals में उसको अपन Golgi bodies या Golgi apparatus कहते हैं.
Okay.
Okay. Now, next are the functions of Golgi apparatus. What is the function of Golgi apparatus? You can see transportation, secretion, storage, synthesis of starch, formation of lysosomes, and lastly, modification and packaging of products of Golgi in vesicles. Now, to understand this function, सबसे पहले diagram पे ध्यान दें.
Yes, sir.
ये आपका सेल है, ये आपका न्यूक्लियस है, ये आपका ER है और ER से बनता है आपका Golgi bodies. What is in general function of a nucleus in a cell?
Sir, it helps in the functioning.
Good. It helps in the functioning of the cell. It is a controller of the cell. Okay.
Yes, sir.
अब nucleus के अंदर जो information होता है, that is what we will study in our part is DNA. Okay. So the one which carries the genetic information. Okay. तो ये जो nucleus के अंदर information होता है, that would be passed. Nucleus किसको connect करता है? To the endoplasmic reticulum. So all this information will go into endoplasmic reticulum and where it will synthesize proteins. So protein synthesis, lipid synthesis, बहुत कुछ बनता है इस ER के अंदर. अब ER detach होके it is pressed और उसी ER से बनता है आपका गोल्गी बॉडीज। अब उस गोल्गी बॉडीज में यू कैन सी ओवर हियर इन द डायग्राम, मैं ग्रीन कलर दिखा रहा हूं, द प्रोडक्ट्स व्हिच आर फॉर्म ओवर हियर। अब ये ग्रीन कलर के प्रोडक्ट्स है जो गोल्गी बॉडीज में बने। व्हाट इज द एक्सट्रीम एंड ऑफ गोल्गी बॉडीज का शेप कैसा है एक्सट्रीम एंड में?
So it is bulged.
Bulge, circular like structures. So all the products which are produced in the ER, ultimately in the Golgi bodies, would be going into this bulge out structures which are known as vesicles, और ये भरने के बाद डिटैच हो जाते हैं।
Yes,
It's a
sir.
simple way to understand. मैंने लास्ट टाइम आपको एग्जांपल दिया था कि मान लो there is a LG फैक्ट्री, तो LG फैक्ट्री मोबाइल बनाती है, मोबाइल बन गए, अब मोबाइल बनने के बाद क्या करना है?
So they transported.
Okay. बनने और ट्रांसपोर्ट के बीच में क्या होता है?
So they are packed.
अच्छा, बनने, पैक और ट्रांसपोर्ट, तो बनने और पैकेज के बीच में कुछ तो होता है।
So
I'll give another example. Okay, you go to a tailor for making a dress.
yes.
आपने ड्रेस दे दिया, ओके, ड्रेस लेके आ गए, पैक करके आ गए, पर उसके बीच में कुछ तो होता है।
So they have to stitch it.
स्टिच किया, वो सब कुछ बन गया, बनने के बाद क्या?
Sir, I'll wear it.
For what purpose?
So for going out.
ट्रायल करोगे ना कि
Yes.
प्रॉपरली बना है कि नहीं। If there is some wrong thing, what will happen?
So I have to go there and then tell them to.
Okay. There are two options. छोटा मोटा एरर है which can be taken care of. Okay, तो वहां पे रिपेयर करेगा, विल से कि ओके अब 2 दिन रख के जाना, मैं वापस दूंगा, वापस बुलाऊंगा।
ठीक है।
नहीं ना, एक छोटा सा बटन निकला है, प्रॉपर लगानी है, तो वो 1-2 मिनट का काम है, विल वेट 5-10 मिनट्स के लिए आप वेट करोगे एंड देन यू विल टेक इट एंड गो।
Yes.
But there is a drastic error in the dress.
Then we have
तो
to.
हाँ, आई विल से मुझे ये नहीं चाहिए, अब वापस बनाओ, वो आपका लुकआउट है, मैंने तो पैसा दे रही थी ना प्रॉपरली।
Yes, sir.
Exactly same thing ध्वनि would be गोल्गी बॉडीज का एक फंक्शन है ट्रांसपोर्ट करना। जो भी चीज उसने बनाई that needs to be transported to the various parts of the cell via vesicles, तो वो एंड में जो बॉल्स बनते हैं उसमें वो पैक होंगे और वो पैक होकर उसको डिटैच करके इट विल बी पास्ड ऑन। अनदर फंक्शन सिक्रीशन, गोल्गी बॉडीज सिक्रेट भी करते हैं सर्टेन प्रोटीन्स, एंजाइम्स एक्सेट्रा, वो बनाते भी और सिक्रेट भी करते हैं इन द गोल्गी बॉडीज। थर्ड, स्टोरेज, अब वो बनाते हैं और वो डिस्पैच करते हैं, तो उसके बीच में वो स्टोर भी तो करेंगे थोड़े टाइम के लिए, जब रिक्वायर्ड नहीं तो इट विल स्टोर, तो वहां पे स्टोरेज लिखा होता है। सिंथेसिस ऑफ स्टार्च, अगेन सिंथेसिस ऑफ स्टार्च, स्पेशल फंक्शन लाइक प्रोटीन सिंथेसिस। अब 2 मेन फंक्शंस है गोल्गी बॉडीज के जो आपके बुक के लास्ट सेकंड लास्ट पॉइंट दिए। फर्स्ट विल टेक द लास्ट वन एंड देन कम टू सेकंड लास्ट। इट हेल्प्स इन मॉडिफिकेशन एंड पैकेजिंग ऑफ प्रोडक्ट्स ऑफ गोल्गी इन वेसिकल्स। अब जो भी प्रोडक्ट गोल्गी बॉडीज ने बनाया, इट्स लाइक अ टेलर ने वो कपड़ा सिलाया, अब वो फाइनल रिहर्सल के लिए, मतलब फाइनल चेकअप करने के लिए आपके पास, आपने उसको ट्राई किया, आपने बोला इट्स ओके, तो वो पैक करेगा। एग्जैक्टली सेम थिंग गोल्गी बॉडीज में वॉट एवर प्रोडक्ट्स आर देयर, दे आर इन अ रॉ स्टेट, उनको मॉडिफाई करना पड़ता है, उनको प्रॉपरली चेंज करना पड़ता है, तब जाके दे आर प्रॉपरली फॉर्म, and then once they are ready to use, they would be packed and dispatched via vesicle. So it brings a very important function, modification and packaging of products in the Golgi, in the vesicles. Am I clear?
Yes, sir.
Okay. अब Golgi body से ही बनते हैं lysosomes, formation of lysosomes. Now what is formation of lysosomes? Can you see the green color?
Yes, sir. So when there is digestive enzymes and
गुड. अगर उसके अंदर कोई सिक्रेटिव सब्सटेंस है लाइक प्रोटीन्स या कुछ भी है बट नॉट इन्वॉल्व इन डाइजेशन then I would refer it simply as vesicles. But if that vesicles have digestive enzymes, then I refer it as your
lysosomes.
lysosomes. Am I clear? So Golgi ER से बनता है Golgi body से बनता है next organelle lysosomes. Okay. Now we go to the next part, lysosomes. What are lysosomes? They are nothing but circular structures. एक ऐसे round structures है जिनके अंदर digestive enzymes are present.
Yes, sir.
You can see the lysosomes are membrane-bound sacs containing digestive enzymes. These enzymes are made by RER. Enzymes RER से बनते हुए packed in Golgi body और Golgi body से जब detach हो जाता है तब वो बन जाता है lysosomes.
So, but you had told me once that Golgi bodies are made from SCR, and when the
Golgi bodies are made from both RER as well as SCR.
okay.
सिर्फ RER सिर्फ SCR से नहीं, दोनों में से कोई भी बन सकता है.
So what about lysosomes? The same thing, right?
No, RER SCR बनेगा Golgi bodies में. तो RER SCR ने जो प्रोटीन बनाए थे वो Golgi bodies में. अब Golgi bodies में से वो प्रोटीन रीचैनलाइज होकर डिस्ट्रीब्यूट होते हैं टू द एंड्स, फार एंड्स. वहां से वो पैक होकर डिटैच करते हैं. जब वो डिटैच करते हैं Golgi bodies से, and if that detached part is having digestive enzymes, तब उसको हम बोलेंगे lysosomes.
Okay.
So ultimately lysosomes कहाँ से आ रहा है? Golgi bodies से. समझ
Yes.
में आया?
Yes, sir.
Okay. Now, some function of the lysosomes. पहला function दिया है digestive bag. Second function दिया है suicidal bags. Now, what is digestive bag? Whatever digestive enzymes are present in the lysosomes, they would simply burst in the cell and they will help in digestion of the organic material or unwanted cellular parts or damaged cell organelles, etc. तो उनको बोलते हैं अपन digestive bag. Am I clear?
Yes, sir.
Okay, simple example. मान लो आपके घर पे you have glass bottle in your hand. Okay, आपके हाथ से glass bottle नीचे गिर गया. What will happen?
That will break.
So what you need to do?
Sir, I have to clean it up.
Good. You will clean it up because it is going to hurt. Is that bottle piece is any use for you? No. It's not exactly same thing. Lysosomes are nothing but like a cleaning agent. वो किसको clean कर रहा है? Damaged body parts, damaged cells, damaged cell organelles. Do we require damaged material?
No, sir.
No. So at that time, lysosomes मान लो इस cell के अंदर ये mitochondria जो damage हो चुका है. It has a damaged part. So what will happen at this point? The lysosomes, ये lysosomes यहाँ पे आएंगे and they'll simply burst open. So all the enzymes will be released at this mitochondria और इस mitochondria को पूरा digest कर लेगा. And that is known as digestive function of lysosomes. Am I clear?
So these lysosomes are formed, and when they have something that is not needed, they will burst it out, and the mitochondria will take it in.
Mitochondria would be clean, मतलब वो degenerate कर लेगा, उसको वो खा लेगा. It will clean it out. Okay, whether it is mitochondria or any cell organelles. I'll give you a proper example also if you want. But wait. Now, second is, अब आपके हाथ में मैंने bottles दिए वापस एक बार. They are not broken. They are very costly और टूटे भी नहीं आपके हाथ में रहते. पागल हो क्या आपको बाहर फेंकने के लिए?
So I didn't understand.
I've given you bottles or the bottles are kept in the room. Okay, the bottles are not broken. They are proper bottles. Will you go and pick it up and throw it out?
No.
No. Is it required?
No, sir.
नहीं, bottle चाहिए. हाँ. But आप अगर करोगे तो पागलपन है, मम्मी डैडी चिल्लाएंगे क्या पागल हुई क्या बॉटल फेंकने के लिए, आया ना।
Yes, sir.
मतलब एग्जैक्टली सेम थिंग. व्हाट इज सुसाइडल बैग्स? लाइसोसोम्स किसको, डज लाइसोसोम ईट?
Cell lysosome eats.
व्हाट पार्ट?
So
वो किसको डिस्ट्रॉय करता है या वो किसको ईट करता है?
the
डैमेज सेल्स, डैमेज सेल ऑर्गेनल्स, अनवांटेड थिंग्स, उनको खा रहा है. ओके, बट सर्टेन टाइम्स में लाइसोसोम्स fail to identify it is damaged or unwanted or it is broken. Okay, वो नॉर्मल ऑर्गेनल्स को डिस्ट्रॉय करता है.
yes,
So
sir.
when it starts destroying its normal organelle, in that case the cell would be affected. तब उसको बोलते हैं सुसाइडल बैग्स, बिकॉज वो खुद ही अपने सेल को मार देता है.
Okay.
Okay, it's like you are playing with a knife. Okay, and then suddenly you put the knife in your stomach itself. उसको सुसाइड ही बोलेंगे ना अपन.
Yes.
Okay, but if you pick it to somebody else तो उसको मर्डर बोलेंगे. वो
Yes.
मर्डर और ये सुसाइड. तो पहले केस में डाइजेस्टिव बैग था, दूसरे केस में वो सुसाइडल बैग.
So, sir, lysosomes can be dangerous also.
Yes, a very simple example of this, not exactly but उसके similar में आता है. Have you heard about your arthritis?
Yes, sir, I have heard about it, but I don't know what it is.
आपके joints के बीच में fluid होता है, वो खराब हो जाता है, it gets reduced. You can give a similar type is lysosomes. तो वो lysosome उसी fluid को destroy कर देता है. तो अपने बॉडी खुद की fluids को destroy कर लेती है. So then there is a friction between the bones and it creates a problem. Okay, just a example, but a proper example just to understand. क्या खुद के सेल जब अपनी खुद की बैंड बजाते हैं, what is the problem? If it is killing a dead, worn out, damaged, not wanted cell, then it is a digestive function.
Yes,
But
sir.
if not, then it is a problematic.
Sir, but why does it do it? Like, why does the lysosome start eating?
Reason because that it has digestive enzymes. So it has failed in its function to identify whether it is required or whether it is not required, whether it is of own part or whether it is a damaged part. तो जिस दिन वो ये मिस आउट कर देता है तब ये प्रॉब्लम क्रिएट होता है.
Okay.
ओके, सेम एग्जांपल इज कैंसर. ओके, अगर सेल डिवाइड करते हैं, ऑल सेल डिवाइड्स, पर वो नॉर्मल कंट्रोल में डिवाइड कर रहे हैं तो अपन नॉर्मल बॉडी है. पर जिस दिन वो नॉर्मल कंट्रोल लूज करते हैं तो उसको क्या बोलते हैं? कैंसर बोलते हैं.
Sir.
ठीक है, कैंसर क्यों होता है? देयर कुड बी 1000 रीजंस ऑफ इट. हो जाता ना. कैन यू प्रिडिक्ट कैंसर होगा आपको? नहीं, कोई प्रिडिक्ट नहीं करता है. एंड इट कैन ऑकर एट एनी पॉइंट. एग्जैक्टली सेम थिंग ओवर हियर. नेक्स्ट इज द mitochondria. A very important organelle next to nucleus is mitochondria. Now you can see the structure of mitochondria. Mitochondria is the largest cell organelle and it is a double membrane structure. दो membrane है, outer membrane and inner membrane. Most unique over there is the mitochondria are different shapes. कोई sausage होता है, कोई elongated होता है, कोई cylindrical होता है, कोई slightly roundish होता है, तो कोई dish shape होता है. अलग अलग shape के होते हैं. But commonly and majoritarily it is sausage shape. I hope sausage पता है. Okay, वो hot dog में होता है ना almost sausage वैसे. So it is rightly round and then slightly bulged out in between, again a part which is bulged out. That is what we say as sausage shape. अपन ने diagram जो draw किया था, what was sausage shape. Okay, now double membrane है. The outer membrane is smooth, but the inner membrane is folded. You can see the diagram. Outer membrane ऐसे होता है. ये smooth है. पर inner membrane कैसे है? It shows finger-like projections.
Yes, sir.
Now, why the finger-like positions are seen?
So because it is having some motion inside it.
It can have.
So it is having some motion inside it. It is
And whenever there is a finger-like position, the only reason for it is it increases the surface area. Just 1 second. The lecture ends at 7:30 now.
So
Okay. Now, जो membrane folding होती है, that is because to increase the surface area. Why surface area? Because mitochondria is the powerhouse of a cell. जितना ज़्यादा surface area होगा, उतना ज़्यादा वो energy create कर पाएगा. So the finger-like position are there to increase the surface area for ATP generation. So what is ATP? Energy currency of your body. तो फंक्शन में भी वही लिखा रहेगा, पावर हाउस ऑफ अ सेल एंड प्रोटीन सिंथेसिस. नाउ व्हाई पावर हाउस एंड प्रोटीन सिंथेसिस? Dhwani, we would continue this in the next lecture. There are only 2 minutes left, so I don't want. अब जब ठीक हो जाओगे, आपका भी concentration से पूरा ही होगा और मेरा यहाँ पे आवाज ज्यादा होगा. So we'll continue it in the next lecture.
yes.
Is it okay?
But I had a doubt.
बोलो.
If mitochondria is the largest cell organelle, so does that mean that it is larger than nucleus?
No, it is as good as nucleus. Need not be as large as nucleus. Okay? It may be smaller than nucleus, or it may be that nucleus is slightly smaller or slightly bigger. But compared to other organelles, mitochondria is bigger.
Yes, sir.
Fine. We'll continue in next lecture. Have a nice day.
So, bye, sir. Thank you.
Bye.
Sir, should
Yeah.
I start this class or should I leave?
Okay. एक second. आपका वापस lecture भी है ना मैथ्स का?
Yes, sir.
So okay, fine. What I can do is I would leave. You be here only. आप switch off करके रखना वीडियो. 5 मिनट के बाद चलोगे.
Yes, sir.
Then have a rest.
Yes, sir. Hello, sir. Sir, I can't hear you. So you haven't connected to the audio.
Donnie, can you hear me?
Yes, sir.
आपने join audio नहीं किया था. Okay, how are you? What happened to you?
Sir, I am having phone.
बोर्ड दिखाइए. ये नहीं. उनको बोर्ड दिखना चाहिए ना. हाँ बस अभी ठीक है. चलो. Yes. Yeah, you can see the board, na?
Yes, sir.
Not till where had we done in the last class?
So you had taught me how to point out 9 point.
But we had finished that exercise. That's all, no?
Yes, sir.
And we were to start with some indices. Remember?
So we were going to start with these. So 1 second.
Just open your volume. We want to start with the exponents.
Yes, sir. Sir, but before that, I wanted to tell you that ma'am taught us with the addition and subtraction, multiplication and division.
What?
So ma'am had started with addition, subtraction, multiplication and division, but I was having difficulty doing that.
Of what? Of what? Addition, multiplication, subtraction, division of what?
So firstly we had to expand it, so I don't know like how to exactly do that.
No, but which chapter are you talking of?
So this one only.
So in that, can you show me the sums which you want me to do?
Yes, sir. So ma'am had given this from NCERT and few of them she had given from herself. So
Show me.
so one of them is exercise 1.5. 1 second. So questions like these.
Yeah, but then we have done these type of questions. If you see exercise 1.4, if you see our volume unsolved exercise 1.4, question number 2. See question number 2, identical sums.
Yes, sir, but what about the division one?
Division 1.
Yes, sir.
That means division 1, okay. Show me, division 1, can you show me the sums that she has done so that I can also give you a similar type of sums?
Yes, sir.
Don't close your book, keep it open so that I can.
So this is one of the sums she had given.
Yeah, so take it up. Oh, this is so simple. This is so simple.
Sir, but she had told us that first we had to write a perfect square.
Sorry.
Sir, she had told us that first we had have to write a perfect square. Like suppose it is written 1250, then I have to write 125 into 10 into 3.
1250 is 125 into 10, na? Why into 3?
So there is the question is root 1250 into 3. So that's why it is 125 into 10 into 3.
So then,
So then
see
we
what
have-
we can do is, I'll tell you what. Now this, when you get the book to me next class, no, that time I can show it to you.
yes, sir.
Right now, let us start with the exponents thing, because otherwise, any specific sum I'll not be able to teach you then.
Yes, sir.
Okay. So we have our laws of exponents. Okay. You can see the board.
Yes, sir.
Now exponents, when I write something like a raised to m, this m here is called as the exponent. Okay.
Yes, sir.
Or it is called as index, or it is called as degree, or it is called as power. It has got different names.
Okay.
And this a down here, this is called as the base.
Yes.
So we come to the first law of indices, which is a raised to m into a raised to n. Now into we can also use dots. That will become a raised to m plus n. When the base is same, the powers get added.
Yes.
Example, I'm writing in red. 3 raised to 2 into 3 raised to 4 will become 3 raised to 2 plus 4,
Yes, sir.
which is 3 raised to 6.
Yes.
Second, a raised to n upon or divided by a raised to n is a raised to n minus n. Bases are same and there is division, the powers will get subtracted.
I had it up.
5 raised to 4 divided by 5 raised to 3 it becomes 5 raised to 4 minus 3, which is 5 raised to 1, which is 5 itself.
So I had it out. In the first one, it is written a raised to m minus, or is it into?
This one,
Yes, sir.
it's into. This dot means into.
Okay.
Third, a raised to m, the whole raised to n is a raised to m into n. That means if I write b squared, the whole cube, this will become b 2 into 3. So it will become b raised to 6.
Okay.
Fourth, a b the whole raised to m is a raised to m into b raised to m. Example, if I write 5x, the whole cube, it becomes 5 cube into x cube. Now 5 cube is 125. So it becomes 125 x cube. Is this clear?
Yes, sir. So I had a doubt. Sorry, it's not a doubt, but so I can't see the second and the third one because of the reflection of the light on the board.
Okay, what I'll do is, I will now better know this.
Yes, sir. Yes, sir. Now it's better.
Now it's better?
Yes,
Okay.
sir.
Okay. Now, the fifth law, a upon b the whole raised to n is A raised to M upon B raised to M. So if I have 2 by 3, the whole cube, that becomes 2 cube upon 3 cube, 8 by 27.
Okay.
Sixth law, A raised to 0 is 1. Any number raised to 0 is 1.
Yes, sir.
Seventh law, A raised to minus M is 1 upon A raised to M. That means 3 raised to minus 2, if I write, it can be written as 1 upon 3 squared.
Okay.
Or if I give you something different, if I write like this, 8 upon 60, the whole raised to minus 2, this I can write as 60 upon 8,
Raised to 2.
the whole raised to 2. Did you understand?
Yes, sir.
Then if I look at some other examples, if I tell you to solve something like this, 64 raised to minus 2 by 3. So what you will do is, 64 first of all can be written as 8 squared or it can be written as 4 cube. Both are correct. But I am writing it as 4 raised to 3 so that I can cancel this 3 and 3. Correct?
Yes, sir.
So this will become 4 raised to minus 2, which is 1 upon 4 raised to 2, which I will write as 1 upon 16.
Okay.
Did you understand?
Yes, sir.
Sure?
Yes, sir. Sir, don't erase it. I'm writing it down.
Sorry?
Sir, don't erase it. I'm writing it down.
I'm not going to erase anything till the end of the lecture. Don't worry.
Yes. Sir,
Yeah.
in the fourth question, it is written AB raised to M.
Yeah.
It is A M raised to M into B raised A raised to M, right?
Yeah. Correct.
had a doubt.
Yeah, tell me.
Sir, why can't I write? So the question 64 raised to minus 2 upon 3. Why can't I write it as this? Like,
What can it as means you want to write it as 8 squared, right?
yes, sir, 8 square, and then I can cancel 2 and minus 2.
No, because see, minus 2 is in the numerator. And
Yes.
when you 8 squared, it means 8 raised to 2 by 1. Can you cancel numerator with numerator?
No, sir.
You can cancel numerator with denominator.
Yes, sir.
Never can you cancel numerator with numerator.
Okay.
Yeah.
Yes, sir. Done.
Done now?
Yes, sir.
Finished all?
Yes, sir.
Okay, now open your volume to exercise, unsolved exercise 1.5.
Yes, sir.
Start doing the questions and you can check your answer because I cannot see it right now. So you just keep checking your answer with the answer key given at the back, okay?
Okay.
And if you are having a doubt, immediately ask me.
Yes, sir. question number 1, 1, 2, 3, all of them are correct.
Very good. Now move on to question number 2.
Yes, sir. The question 2 also is correct.
All the 4 sums?
Sir.
Good. Now move on to question 3.
Yes, sir. And the second question, the third second question, sub question second, I had doubt.
Which one? Can you read it for me?
Yes, sir. So 1/3 raised to 3 bracket and outside.
7.
Yes, sir. So I got the answer as 1/21.
1/3 raised to 21.
Yes, sir. So I multiplied both the exponents. I got 21, 1/3 raised to 21, but behind it's written 3 minus 21. So, sir, is it okay if I write it as 1/3 raised to
Yeah,
21?
generally what happens is generally we take everything into the numerator. Generally.
Okay.
So it is better, but even if you leave it as 1/3 raised to 21, you will not get wrong.
Okay. Yes, sir. So how do I solve the third one?
Which third? 11 raised to half, that one.
So,
That's some, no?
yes, sir.
Okay. See, what we are going to use is, we are going to use this law, the second one. 11 raised to half upon 11 raised to 1/4. That becomes 11 raised to half minus 1/4.
Yes, sir.
LCM is 4, so 2 minus 1. So that will become 11 raised to 1/4.
Okay, but sir, how did you take 11?
Sorry?
So how did you take 11 up?
See, it is like this, no? A raised to m divided by a raised to n. It automatically becomes a raised to m minus n, second law.
Yes, sir. Okay.
Understood?
Can you explain the fourth sum also?
Fourth sum,
Yes, sir.
7 raised to half into 8 raised to half. Now see, this is like this, fourth law.
Okay.
Powers are same, correct? So 7 raised to half into 8 raised to half. I can write it as 7 into 8 the whole raised to half, yes or no?
Yes, sir.
So your answer is 56 raised to half.
Okay.
This is the reverse of this fourth law.
Yes, sir. Got it. Sir,
Yeah,
sir, how will I do the fourth sum?
fourth sum. Okay, I will show you the fourth sum on the board. Okay. Now, if you see, both the powers are negative, right?
Yes, sir.
So I will take this 216 up and write this as positive power, like I did. I gave you the last law. Remember that?
Yes, sir.
Same way I'm going to do this here 27 4 by 3. Now 216 is the cube of 6 and 27 is the cube of 3. Correct? Remove this. Yes or no?
Yes.
Now this 3 and this 3 will get cancelled, this 3 and this 3 will get cancelled. So I'm left with 6 squared divided by 3 raised to 4. Correct?
Yes, sir.
6 squared is 6 into 6. 3 raised to 4 is 3 written 4 times.
Okay.
Ones are, twos are. Ones are, twos are. My answer is 4 by 9.
Okay, sir.
Clear?
Yes, sir. Uh, I can't understand the fifth question, although I'm halfway through. So can you explain it once?
What are you saying? What are you saying? I can't understand.
So can you explain me the fifth question?
Fifth one. Fifth one also. Can you not try to change the powers which are negative, make them positive? Can you try that first?
I did that.
Still did not get the answer?
No, sir. So actually, I'm halfway through, but it would be better if you would.
Oh, I'll show you. I'll show you. I'll show you. So now first I do this 4
Yes, sir.
squared. Okay?
Yes.
3. This I'll write as 2 cube, the whole raised to 2 by 3. Any number raised to 0 is 1.
4 will become 2 square, right?
Then this I'll write as 16 by 9, the whole raised to half. Okay? Now 4 square is 16. This 3 and 3 gets cancelled. 2 squared is 4. This becomes 4 by 3, the whole square, the whole raised to half. Did you understand this?
No, sir. So can you repeat?
What is 16?
4 square.
What is 9?
3 square.
So can I not write as 4 upon 3 the whole square?
Yes, sir.
So that this 2 and 2 will get cancelled. So it becomes 16 minus 12 plus 4 by 3. 16 minus 12 is 4. Understood?
Yes, sir. Yes, sir. Done.
Can do the next one also, sixth one.
I can't understand this question either.
Okay.
So I have done the 125 upon 64 raised to 2 upon 3.
Wait. 64 upon 125 raised to minus 2 by 3 plus 1 upon 256 upon 625 the whole raised to 1 by 4 plus root of 25 upon cube root of 64. So see this I will 125 upon 64 the whole raised to 2 by 3. This I can take it up 625 upon 256 the whole raised to 1 by 4. Yes or no? Plus, what is the square root of 25? 5. What is the cube root of 64? 4.
So, but I had a doubt.
Huh?
I had a doubt.
On what?
So 625 upon 256, how did it like take it above?
When I take it above, it will become minus.
Yes, sir.
But for minus, I made it ulta. Instead of writing 256 upon 625, I wrote 625 upon 256 in the same step.
Yes, sir.
Now this is 5 by 4, the whole cube, the whole raised to 2 by 3. This will become 5 by 4, the whole raised to 4, the whole raised to 1 by 4, plus 5 by 4. Did you understand this step?
Yes, sir.
Now, this 3 and this 3 will get cancelled. This 4 and this 4 will get cancelled. So it is 5 by 4 the whole square, which will become 25 by 16 plus 5 by 4 plus 5 by 4. My LCM is 16. So this is 25 plus 20 plus 20. 65 by 16. Answer. You understood?
Yes, sir. Yes, sir, done.
Done. Then you can try those homework sums for homework. Okay?
Yes, sir.
There are only 2 questions. Then next class, if you are feeling fine,
Yes, sir.
feeling okay, when you come for the next class, get your school notebook.
Yes, sir.
So that whatever the questions that you are saying your teacher is making to do, we can do those.
Yes, sir.
Okay? Take care of your health. Bye. Good night and God bless you.
Bye, sir. Thank you.
Sukas,